Oh, great. The mic is working. I managed to knock it off my lapel. We're good here. How do I switch to the screen? Thank you. It is just a tremendous pleasure to be back here in Barcelona, which is not only a wonderful intellectual center, but just an amazing city. And I actually got inspired to try and put a lot of new stuff into this talk. And this is joint work with a variety of members of my lab and Maria Samsonova's lab in Russia, in St. Petersburg. And so a large part of this talk is actually not going to be about a network. It will be about a single node of the network. And the emphasis will be on getting the input-output relations of this node right at a fine scale controlled by DNA sequence. And this has to do with understanding the second half of the genetic code. That is to say, the regulatory code as opposed to the structural code for proteins. And time permitting, I will go on and say some things about the whole network in terms of dynamical systems and the dynamical basis of decanalization in mutants. So let me progress first to the single node, even skipped. It's a node in the network of segmentation genes. This is a slide that was made by my student many years ago. And essentially what happens in the early embryo of Drosophila, these are embryos that have been stained with antibodies. Each dot is a nucleus. Anterior is to the left, dorsal is up. And the brightness of each nucleus is proportional to the concentration of some particular protein in it. And so what happens early in Drosophila development is that asymmetric initial conditions, of which I'll have a lot to say in particular about bicoid and a little bit about caudal, give rise to expression of gap genes in bands of nuclei about 10 or 20 nuclei wide. And the expression of these gap genes drives the expression of pair rule genes in pattern of 7 repeating stripes. This is the pattern of even skipped, and I'll have quite a bit more to say about even skipped in the first part of the talk. And then ultimately, even skipped together with other pair rule genes gives rise to expression of engrailed and wingless, which actually create the segmental borders. So in the first part of the talk, I'll be talking about Eve in some detail. And then in the second part, I'll be talking about, about the network of gap genes that controls the expression of Eve. So to make this pattern of 7 stripes actually requires quite a bit of DNA. So the entire Eve gene is 16.5 kilobases. That's known from embryonic rescue experiments. And of that 16.5 kilobases, only 1.5 kilobases is transcript. The rest is not junk. It is absolutely essential regulatory DNA. Now, the classic picture of the DNA, the 14 kilobases or so needed to make the 7 stripe pattern is that you can divide it up into enhancers. And enhancers are little isolated pieces of DNA that drive a part of the pattern. For example, this is the minimal stripe element for stripe 2. In isolation, it will drive this stripe. And typically, these enhancers contain clusters of binding sites. But there are many, many details of how these enhancers work that are unknown. And in particular, if you take this enhancer for stripe 2 and move it immediately adjacent to the enhancer for stripe 3, you get a completely different pattern. And we'll be talking about how to exploit that fact a little later in the talk. But first I want to talk about a really serious problem that comes up in thinking about the evolution of enhancers, which is that function is conserved even when the regulatory sequence becomes extremely diverged. And so one example of that you can see from a classic study by Misha Ludwig and Marty Kreitman, which is even among enhancers from species that diverged over 10 million years ago, Pseudoobscura, Erecta, Jacoba, and Melanogaster, considerable sequence divergence has occurred, but all of these species have enhancers which, if placed in Melanogaster, this is Melanogaster on the top, Jacoba, Erecta, Pseudoobscura, will all give stripes in Melanogaster. And in fact, it's even a more severe problem than that. This is a Drosophila melanogaster. This is Sepsidae synipsia. These are flies from a completely different order, the sepsids. They eat cow dung. They have very, very diverged sequences. Nevertheless, they have an even skip gene too. Very homologous at the protein coding level. But here's melanogaster, synipsia, and putris. These are blastids. So what you can see here is synipsia and putris have considerable homology. But the homology between the sepsids and melanogaster is almost nothing. The only stretches of homologous DNA specify particular binding sites. And yet, amazingly enough, in a brilliant experiment by Emily Hare and Michael Eisen, if you take the synipsia stripe 2 enhancer or the one from putris and place it in melanogaster, you get a stripe 2, and it is almost in the same location as the native stripe 2 driven by the stripe 2 enhancer from melanogaster. And these are really quite diverged. So we find it useful to consider the minimum edit distance as a measure of distance. That's the minimum number of insertions, deletions, or substitutions you need to alter one sequence to the other. These red bars are the edit distance. And you can see there's some like some species like simulins that are very close to melanogaster. Jacoba and Erecta are considerably far away, 26 or 25% changes. And Pseudoobscura is even further. And then the sepsids are extremely diverged. So this is a really basic and fundamental problem, which is how is it that you're able to conserve function when the DNA sequence is not being conserved at all? Okay, this is a very basic problem in evolutionary biology. So how to attack it? Well, there's two tools. One is a high-resolution One is a high-resolution transcription data set that we obtain from confocal images. This data set is downloadable. And this gives you a numerical atlas of the transcription factors that control expression. Because it's not just sequence that controls expression, it's transcription factors that bind to the DNA. So if you want to have a quantitative model, you need both things. You need the sequence, and you need to have precise data about transcription factor concentration. Now what's happening with even skipped is that the 7 stripes of Eve expression are being generated through activation by bicoid and caudal and other factors such as dstat and repression by these very broad gap domains. And there's some very tricky points here because you can see that this repression can't be absolute because even though it's known that Krupel sets the posterior border of stripe 2 by repression, it's certainly not silencing Eve because since Krupel is so broad, that would cause a big zone of non-expressing cells here. So in fact, this happens by a short-range repression process, they'll have a few more things to say later. And then with these transcription factors, you then can insert chromosomally integrated reporters into the melanogaster genome and read off their expression. By the way, these little points are actually nascent transcripts. So here you see 2 dots. That's from the 2 copies of the Eve gene. And these are coming off the genomic DNA literally as we watch. And we segment these, and we can register them to the protein pattern. We also stain for Eve. And so this gives us a very precise data set that essentially amounts to using the Drosophila embryo as a microarray. But we don't have to pay tens of thousands of dollars for our array. We just feed the mother fly cornmeal and molasses. And for some of you other geneticists, they also like to eat yeast. Now, we also want to make a quantitative model of this control process. And so there's 2 design principles, which essentially amount to, if you actually have scientific knowledge of what's going on, use it, and if you don't, guess. So the one thing that's well understood reasonably well are protein-DNA interactions. If you know the concentrations of transcription factors and you know the DNA sequence, you can calculate the configuration of bound proteins for any set of concentrations of TFs using thermodynamics. And I must point out as a sort of a side comment, people sometimes talk about thermodynamic transcription models. Really, there's no such thing because transcription is a kinetic process. But you can calculate the configurations from thermodynamics, and then you're left with a really fundamental problem, because the great big unsolved issue, indeed, maybe the skeleton in the closet of all of genomics, is that nobody knows the rules that say whether a particular configuration of bound factors is activating or repressing. So we try to guess them. These are the protein-protein interactions, that is to say the regulatory mechanisms, and we try to figure them out by phenomenology, which is just careful guessing, keeping in mind 2 empirical points. So one is that biological function resides in multiple binding sites. I'm not going to give you a chemical explanation of why that is. It's just an observed fact. And the second thing is that activators can have greater than multiplicative synergy. So you could add 1 activator, it would increase transcription by a factor of 3. You'd add another one by itself, would have increased transcription by a factor of 5. The 2 together would not increase transcription by 8. That would be additive, or by 15. They might increase it by 30 or 50. Now, another reason that transcription is hard to understand is that many regulatory mechanisms operate at once. And that makes progress with models slow because you have to add the mechanisms one at a time, and that's not an easy business. You have to think carefully, look at experiments, try and embody other people's experimental results with mathematical equations. But right at the moment, we have 7 mechanisms in the model: proteins binding to DNA, competition for bound sites, cooperative binding, which is only implemented for bicoid. There's very good evidence that bicoid can cooperatively bind with a range of about 60 base pairs. And the geometric picture you should have here is a linear molecule of DNA with various proteins bound to it that are interacting. And of course, we believe that there's no action at a distance, that the DNA folds back, but we don't try to incorporate the 3-dimensional structure into the model. So one very key thing is short-range repression by quenching. That is to say, a repressor or a set of repressors can stop an activator from acting over a range of about 150 base pairs. Similarly, repressors, these short-range repressors, can be turned into activators by certain proteins that bind within 150 base pairs of them. Okay, there appear to be 2 co-activators in this system. bicoid and caudal. And I don't think it's accidental that this range is on the order of the size of a nucleosome. We also consider direct repression and activation by a diffusion-limited Arrhenius rate law, which is essentially the simplest chemical description of a catalyst, and this is fundamentally a catalytic or enzymatic reaction. Now, the next slide is a little scary, but don't be scared. I like to show the full set of equations. Okay, now these look really ugly. The indices are sort of like little bits of hair growing on things. But the first thing I want to say is this is essentially just a big function. That is to say, it says what the transcription rate is as a function of concentrations of transcription factors. Those are these V's, and also as a function of DNA sequence. So this top line is where we use positional weight matrices to calculate equilibrium binding constants from sequence. Okay, this orange square is the thermodynamic calculation. Here is the protein-protein interactions. And essentially what we're doing is we're adding up activators where we turn some of them off by repression. I'll come back to equation 7 in a minute. And we turn some repressors into activators by coactivation. Here in equation 6, we add up the activators. And then this term here, which essentially is a sigmoid threshold function, this embodies the idea of an Arrhenius rate law that is diffusion limited. And just to give you a little bit of the flavor of the phenomenology, this term here is where the short-range repressors act. And so we take every activator. These F's describe the fractional occupancy of a binding site. That's a number that ranges from 0 to 1. And for each activator site, we convolve it with the short-range repressors. They have this occupancy FQ. Notice there's a big product here that looks like 1 minus 3 factors. This is the strength of the repressor. This is the occupancy of its site. And this controls the range. So this function Q of D is 1 for 100 base pairs or less, and then it continuously goes to 0 at 150 base pairs. And the way we work in the fact that you need multiple sites to get biological function is by this factor. So for example, in a 2006 study with Hilde Janssens, it turned out that most activators interacted with about 6 or 8 repressors that were in range. Typically, this factor was about 0.6 for each repressor. And so 0.6 is not a lot of repression. But 0.6 to the 6th power is pretty strong repression. So that's an example of the phenomenology. Now there's various parameters here that we have to determine from data, such as the strengths of these various effects, the strengths of the activators. And so we do this by a large-scale optimization. Each transcription factor has about 2 to 5 free parameters. And we obtain them by a least squares fit to expression data, which we perform by a nonlinear optimization technique called simulated annealing, which I don't have time to discuss today, although it in itself is a very interesting topic. Now, the positional weight matrices come from independent outside data. That means the model is also parameterized by DNA sequence. And I'll comment in passing that some of my friends who do physics, they're very happy with models that don't have parameters. But if you're looking at genomes, of course you're going to have a lot of parameters, because what is a genome but a giant pile of parameters? Can't get around it. Now, I said I would provide you with a few answers, and I'm going to be sort of looking at the clock during this part. So we'll see how many answers I can squeeze in before we get to the dynamical systems. But the fundamental starting point for a lot of this work was something that Aram Kim did. Now, this actually was an animation, and it's been frozen in place in a slightly ugly way. But what I want to point out to you, this is the dashed lines are quantitative data, and the red line is a model fit. And so if you don't see the quantitative data in some of these panels, it's because the overlap is so close that the two lines are coextensive. So the key point is that it was shown a long time ago by Stephen Small that you can put this enhancer for stripe 2 and the enhancer for stripe 3 within as little as 300 base pairs of each other, and you got essentially normal expression. This is stripe 2, stripe 3, and stripe 7. But if you remove this 300 base pairs, you've got a qualitatively altered pattern where stripe 2 and stripe 3 were fused, and you can do it in the opposite order. If you do it in the normal order, the expression of stripe 2 is vastly increased. If you do it in the opposite order, the expression of stripe 2 is greatly reduced. And these are such small perturbations at the sequence level that cause such big changes in expression. We reasoned that if we could model those, they would seriously constrain the possible rules of regulatory sequence. And we threw in other data that we had for the enhancers separately, for the enhancer for stripe 2, or for 3, which also drives 7. And I'm not going to talk about this work a lot because it's now published in PLOS Genetics, but I do want to show perhaps my favorite result from that paper, which is reassuring in terms of the strength of the model. Okay, so using only data from these rearrangements of the Drosophila Eve promoter, we then took all sorts of other DNA sequences that were out there and checked what the model predicted. So in fact, we predict a stripe from Synapsia. This is Eve's stripe 2. This is the stripe we predict. Notice it's a little posterior to Eve's stripe 2. This is what Heredal found. But in fact, what they found from measuring against the embryo, this is the position of the borders of the stripe in melanogaster. They're measuring percent egg length from the posterior pole. 63 and 57, okay? Notice that they found the stripe to be expressed a little bit, essentially about 1 or 2 nuclei posterior from the native location. Okay, that's essentially what you're seeing here. We exaggerate the size of the shift by maybe 1 or 2 nuclei. So this is not up to the level of quantum electrodynamics, but in the context of biology, this isn't bad. And these are basically non-homologous stripes. Now, we think we understand what's going on mechanistically here, but I don't want to comment about it today because we're checking it experimentally. And instead, I want to talk about some other parts of the problem having to do with the expression of stripe 2 from different Drosophila species, erecta, pseudoobscura, and yacuba, where the model also recapitulates concerned gene expression pattern. Now, the nice thing about having a model is that once you get something right, a prediction or data, you can go look inside the model and understand in detail what's going on. And so what I want to do is talk about 1 and possibly 2 cases of analysis of what was happening evolutionarily with these enhancers. So the first one is an analysis of compensatory evolution, which is the resurrection of ancestral enhancers. And this is work with Carlos Martinez and Josh Ress. So one of the problems here is it takes a long time to synthesize and test these enhancers. So we actually started this project a long time ago with a slightly more primitive form of the model. And we assumed that the various enhancers from different species all drove exactly the same expression. Now, what we then did with some help from Josh is we did a Bayesian reconstruction of the ancestral sequences. And essentially the way you do this is you know the various transition probabilities for each base pair. And if you compare the sequences, you can essentially construct a Bayesian network that will give you the posterior probability of various ancestral sequences of the ancestor. Okay? So this is a Simulans Seychellia ancestor. And the Bayesian methods give various different posterior probabilities for these different sequences. But I emphasize that what the Bayesian methods do has absolutely nothing to do with the function of the DNA. It's entirely based on sequence and the probability of various kinds of mutations and of substitutions and indels. So one question that one can ask is are in fact all of these various possible reconstructions, do they give the same expression? And in fact, what's shown on the horizontal axis of this graph is essentially the root mean squared deviation of a model that uses each of these sequences compared to the actual expression data. And so what we decided to do was to resurrect the ancestral enhancer. Marty called this the Hopeful Monsters Project, but I like Jurassic enhancers better. And express them in melanogaster and see what their expression was. Now, you know, I have to be upfront with you. These two cases worked. This was the melanogaster Simulans Seychellia common ancestor. This one down here, which was the Erecta jacoba common ancestor, for various reasons did not work. We thought that this one would give low expression and this one would give high, this would give low expression and this would give high. But in fact, we got the opposite result. But nevertheless, these worked very nicely. And in a moment, I'll show you what their evolutionary implications were. Once we had constructed the various resurrected enhancers, we then obtained quantitative data from them, which are shown here. This is Simulans Seychellia, melanogaster Simulans Seychellia, and we fit them to the same model from the 2013 paper that predicted the sepsids. And we got a very good fit to data. And then using this fit to data as our evolutionary model, we were able to do some things that you really cannot do by analyzing extant species. So I mentioned that you can look inside the model and see what's going on. So one thing is that the anterior border of the stripe is set by repression from giant. And the stripe itself is activated by bicoid and hunchback. So in fact, there is compensatory evolution going on between the repressors and the activators. And you can sort of see it in a very stochastic way here on this graph, because you have the activation on the vertical axis, repression on the horizontal axis. This is the ancestor, okay? Towards melanogaster, both repression and activation go down. But as you go to the SimSec ancestor, both activation and repression have gone up, and evolution is stochastic. It goes back down again for Seychellia. So this you really need resurrected enhancers for. You can also go back and look at this over a much wider phylogenetic distance with enhancers from many extant species. And in fact, this pattern of compensatory balance extends through the Drosophilidae. And so now that we have a much more precise model, we're going to extend these studies and resurrect enhancers that go further back. Now, this was in some sense a coarse-grained study. I didn't discuss particular sequence, particular binding sites or sequence elements. You can also do a fine-scale analysis of active sites. And in this analysis, look, one of the problems you have that's really confused people analyzing these promoters is there's so many binding sites. Here's the stripe 2 enhancer. You can see there's scads of binding sites all around it, and you really don't know which ones are essential. So what Aram did was he looked at the top 80% contributors to gene expression. For each of those, he looked at the regulatory sites that were affecting them in any way. He discarded any if they were essentially providing no activating effect. And then the result was that you could actually look at a much simpler problem, which was a set of functional groups. In this case, these are the binding sites that control the anterior border of stripe 2. And so once you know that, suddenly things look a lot simpler. There's two activators here. One is hunchback 3 co-activated by bicoid. The other is hunchback 2. And in fact, these are two overlapping functional units. And you can look at what sort of expression they control. Okay? This is not the expression. This is essentially the summed activation. But what you can see here is that the hunchback 3 site actually gives you a stripe. The hunchback 2 site, which is this red, essentially does not give you the spatially delimited expression, but it boosts everything. Okay? And if you've worked with these enhancers, this is an effect you see over and over again, where you'll have something weak that makes a spatial pattern. You add a piece of DNA to it that does nothing by itself, and it just really potentiates expression from the other element. And I don't think I'm going to go over these slides a little rapidly, but once you have these functional groups in hand, you can make a very careful mapping between species that you can't do by sequence alone. So the binding site sequence is fairly close among these species. The coactivation pattern is conserved. So is the competition and the short-range quenching. And so with these points in hand, you can actually make homologies between the function of binding sites that are not apparent from just looking at the DNA sequence. You need this extra functional information to do that. And once you have that, you can actually make a comparison of conserved and non-conserved interactions among the sites. And the result from that is that there are conserved interactions, but they do not support conserved expression. Okay? This is what's conserved between melanogaster and Yakuba will just make this feeble little stripe. You can do a little bit better in pseudo-obscura, but in order to maintain the functional conservation, you need to add some new interactions to build up the conserved function. And I could say more about this. This is being written up for publication. This is, in fact, showing you what the new interactions are. It's essentially, It's essentially what you have in Yakuba is because of the shift of bicoid sites and the deletion of bicoid -1, you get a new cooperative interaction here between bicoids. And that strongly coactivates this hunchback over here. And that's what brings up this feeble expression to a conserved level. Now, at this point, I'm going to shift gears very sharply. I want to talk about the network as a whole with many nodes and the gap gene system. So, a long time ago in 1942, Conrad Waddington noticed two things. One is that tissue types are discrete. They don't form a continuous blend. And the other thing he noticed, which is something all geneticists are aware of, is that the phenotype of animals is variable in mutants but constant in wild type. So this is a first instar Drosophila larva, a wild type with its 8 abdominal segments. And this is Krupel minus. And you can see even at the cuticular level, there's considerable variation in the phenotype. This is from a classic paper by Eric Wishaus back in the 80s. And from that, Waddington predicted that developmental reactions, as they occur in organisms submitted to natural selection, are in general canalized. That is to say, they're adjusted so as to bring about one definite end result, regardless of minor variations in conditions during the course of the reaction. Now, Waddington illustrated this by these beautiful pictures of river valleys, the idea being that this is the state of the embryo, and if some perturbation sloshes things up to the side, it will go back down to the bottom of the valley. As Jordy said, you can use the noise or lose it. So this in a developmental system is in the context of losing it. Now, at the time Waddington made these pictures, there was no math description of canalization. That was devised in the 1960s by René Thom. And the basic idea was that there were attracting sets. And here with this audience, I don't really have to go through attractors in detail. They can be points where the state approaches from all directions. They could also be manifolds or paths. This is an equilibrium point that's a saddle. It has trajectories coming in along one axis. So in this case, it's a saddle with one negative eigenvalue and one positive eigenvalue. And in this case, the unstable manifolds created by the positive eigenvalue, which are unstable with respect to the equilibrium, nevertheless are attractors in the sense that surrounding trajectories tend to approach them. Now, we've published some things on canalization, but today I want to talk about decanalization. Why? Okay, the phenotype of animals is variable in mutants, but constant in wild type. How does that come about? Well, first of all, let me give you a specific example at the level of gene expression. So this is from a paper that was published earlier this year, back to back with a paper on Talis from Yogi's lab. This is the wild type gap pattern over various times following the 13th nuclear division. And this is anterior to the left, posterior to the right. And what you can see is that you can see that the gap gene patterns increase in amplitude over time. They get slightly narrower. In Krupel minus, they're really severely disturbed. I mean, their levels are much lower, and there's a whole bunch of disturbances here, which I'm not going to go into in detail. I will comment, though, that in fact, this posterior giant pattern here, this just shows some data and some modeling results that I'll get back to. Okay, this posterior giant domain, which has moved more anterior than it is in wild type, and CNRPS has gone down almost to 0. This posterior wild type, this posterior giant domain is decanalized. So this is the variance of its border, which increases by about 50% in Krupel minus compared to wild type, and the maximum of the posterior domain, which actually fluctuates quite a bit, about 70% more in Krupel minus than in wild type. So in fact, Krupel does become decanalized at the gene expression level. Now, in order to see why, we need to introduce another idea from dynamical systems, which is the idea of a bifurcation. And this is a qualitative change in the dynamics that frequently, but not always, comes about by the merging of two equilibria, and they will annihilate. Okay, so here we have an attracting point or a node. Here we have a saddle. As the two approach each other, notice geometrically, the flow from here is to the right. The flow here is to the right. Here the flow is to the left. So as these 2 points merge, it's just geometrically inevitable that this stable point will disappear. And by the way, notice that not only is the point attractor disappearing, but the manifolds coming in to the saddle, or if it had the opposite polarity going out from the saddle, also disappear. Okay, so when a bifurcation happens, you can see really big changes in the dynamics. Now, what kind of model are we going to use for this? Well, consider the sequence level model that I showed you before. And remember, all the activation is being added up, okay? It gets added up to M, and there's all kinds of complicated stuff. This is the 2006 model in a slightly different notation than I showed you in the scary model slide. All this complicated stuff is all going into U of V, this QM minus theta. All sorts of complicated stuff. So what if we take this and do a Taylor expansion? So if we do a Taylor expansion to first order around some concentration point V0, these are the concentrations of regulators here. And so we'll have a constant that depends on V0 plus this linear term. And if we collect together all the terms that involve a V0, and we write them as H, that will be U of V0 minus these first derivatives of U with respect to V. This we can write as H. Excuse me, this will be H, and then we can write these sensitivity coefficients here that don't depend on V0. We could write them as T. And actually, if you do that, that approximation gives a model that we've been working with for a long time. This G is a threshold function that looks like this. So there's linear stuff in here, but the equations as a whole are nonlinear. This is synthesis of proteins up here. We add diffusion for transport between adjacent nuclei and first order decay. And we've done a lot of stuff with this model since 1991. And it has many, many advantages. It actually has one problem. As it turns out, these equations, although they're quite predictive for various perturbations of wild type, they fail completely for null mutants. And it took us a long time and a lot of patience to convince ourselves of that. And then we just didn't know what to do. So I didn't talk about this much in public. Because you usually want to talk about your successes. But actually, there's a way to deal with this problem. Because now we know that this model is all part of a Taylor expansion. And if your first order Taylor expansion doesn't work, you can add higher order terms. So that's what we did. Now, ultimately, we'd like to get these second order terms by looking at DNA sequence in some way. There's some problems with that. Because if you think about the previous model, the application of the chain rule for writing down coefficients of this Taylor series is really kind of insane. And you cannot even do any of these calculations without symbolic manipulation programs like Mathematica or Maple. But we think we can make some progress with that. However, in the short term, we want to do something much more simple, which is we want to fit these equations to both wild type and Kruppel minus. And we're going to represent the mutants by setting the synthesis, maximum synthesis rate, R Kruppel, to 0 for the mutants. Now, the tricky thing is that there's an explosion of n cubed parameters here, where n is 4, 5, or 6. That's pretty scary. We don't like that. So we're hoping that we can make this higher order matrix TABC as sparse as possible. Okay, we'd like to make it sparse by doing some beautiful statistical analysis of sequence, but for this first time, we just did it by trial and error. And it turns out that as few as 3 non-zero TABCs, how many minutes? 5? What a relief, I'm almost there. As few as 3 of those TABCs will work. And so you can construct a model. Finally, everyone who's been in my lab has tried to model mutants, and we've tried all kinds of tricks. This one actually worked. This is work with Kostya Kozlov in St. Petersburg and Lena Panok in Chicago. And so you can actually, to a pretty good degree of accuracy, model both wild type and Kruppel minus. Okay, you can see the model is these smooth lines, the jaggedy lines are the data. And the 3 non-zero higher order interactions are hunchback bicoid, Kruppel, and Kruppel-Knirps. And finally, how is it that things get decanalized? So in order to do a dynamical analysis, we fit the average patterns of the gap genes to data using a single individual bicoid pattern. We're interested in canalization with respect to variations in bicoid because we know from previous work that the gap gene system filters and reduces the variance in the early bicoid pattern. So we do the fit on average in the individual patterns. Then we run the full model with 88 individual bicoid profiles, check for canalization in wild type, decanalization in mutants. We then short the equations by turning off diffusion. And then we have, instead of 232 coupled equations, because you have, they're coupled by diffusion, you get 58 sets of 4 coupled equations. And then we're going to turn down the Kruppel synthesis rate continuously from wild type values to a null mutant value in a way that's actually a dynamical representation of an allelic series in genetics. And the results are shown here. These are the 88 individual bicoid gradients that we run through the model. This is wild type. This is Kruppel. Okay, you can see that these are, in fact, these domains are canalized. Okay, in the mutant, they become severely decanalized. Okay, as happens in reality, Knirps goes to 0, giant moves towards the anterior. Now, in the absence of diffusion, they get pretty jaggedy, but you can see the decanalization happening. Now, if you look at the dynamics, you can make phase portraits. And I'd like you to draw your attention to this phase portrait here. Okay, a phase portrait is a picture of the state space with the trajectories the system has represented by these lines. And the green lines are the biologically relevant part. And notice that these various trajectories, okay, we happen to be at 59% egg length here, measured from the anterior pole. These various trajectories are heading over for the giant axis, but as they approach, they're drawn to this unstable manifold U4 that emanates from this saddle, S13. And notice how they come together here. Okay, this is in wild type. Now, what about in mutants? Well, in wild type, okay, here they are from high, medium, and low bicoid, approaching the giant axis. And even though they have different trajectories with respect to Knirps, there's a very low range of giant here. In the mutants, okay, in fact, they end up executing a much higher range of giant. This is the dynamics of decanalization. And the reason that it happens is that S13 gets destroyed in a bifurcation. Here we're turning down the strength of Kruppel from wild type values to null. Okay, S13 annihilates with S22. And once it goes away, that unstable manifold that is emanating from it goes away too. And that, in fact, is what causes the decanalization. So here you can see the patterns in the shorted model at point A, B, C, D. Okay, A, B, C, D. After D, the unstable manifold is gone, and you completely lose canalization. So it's this loss of the unstable manifold that leads to decanalization in mutants. And here is the Kreitman lab. Marty Kreitman and I have merged our laboratory operations. I'll just show you very quickly. Aram Kim. This is Manu. He did some previous work on canalization. Pangyao Jiang, Carlos Martinez, Marty Kreitman. Here is Lena Panok, who made that discovery about decanalization in Kruppel minus. So if you're all still alive after hearing that, I want to thank you very, very much for listening. And if I have a few microseconds left, I can even answer some questions. Thank you so much.